Okay, next up we have Megan Rœa, who's from the National Institute for Math and Biological Synthesis. We have a round of lightning talks.
All right, so hopefully you can see me over the podium because I can't really see anything else. So as you guys have been hearing a lot about today, I'm going to be talking to you about coevolution. So coevolution is one mechanism by which evolutionary processes can shape species interactions. Coevolution describes instances where there's two or more species reciprocally altering each other's evolutionary pathways. And it's important to note that it's most likely to occur for species that are engaged in closely related processes. Ectomycorrhizal fungi form closely related processes with plants. So EMF, or as I'm going to call them today EMF, are characterized by the exchange of soil nutrients, primarily nitrogen and water, for plant carbon in the form of photosynthate. They only form association with 2% of all land plants, but these are primarily woody plants, and they're extremely common in temperate forests as they form associations with pines, oaks, willows, firs, beeches, and birches. And I'm going to note here that pines are obligately ectomycorrhizal, so you will not find pines without ectomycorrhizae, and they represent one of the oldest and most diverse plant associations. And while it's widely accepted that coevolution played a key role in early evolution of these mycorrhizal interactions, its current role in mycorrhizal relationships is an area of much debate. While some researchers really support the idea of ongoing coevolution between mycorrhizal fungi and their hosts, others argue that really we're just seeing changes in response to abiotic pressures, and we need more information in order to really understand what's going on. So in order to do this, we really need data from experiments that are directly estimating selection on plants and their mycorrhizal traits. So to get at this sort of question, in May of 2013, I planted a common garden with 12,000 plants from 43 different families of Monterey pine, or Pinus radiata. Pinus radiata is an economically important species as it's planted worldwide for timber, but it's unique in that it's been restricted to only 5 locations in its native range, 3 on the coast of California and 2 on islands off Baja. This isolation has led these populations to diverge genetically. So what I did for this experiment was bring those plants back to one location, Cambria, California, and planted plants from the 5 main populations, as well as crosses of those populations, which were established as timber demography plots in the 60s. So I took seed from those trees and planted them all out. I had 12,000 plants, and I want to note that this was planted during the hottest and driest summer on record, or as I'm going to call it, an extreme selection event. And this sort of experimental design allowed me to assess 2 main questions. One, how is selection altering plant traits from both a plant fitness model point of view and a fungal fitness model point of view, and what form is mycorrhizal mediated selection taking? So, hitting it quick, if we talk about selection, or how selection altering plant traits from the plant fitness model point of view, we see that diameter and relative growth rate are both are both experiencing increased selection, while root to shoot ratio and specific loop length are both experiencing decreased total selection. And if we look at what's happening from the fungal point of view, we see similar patterns in which both diameter and relative growth rate are both also experiencing increased selection, while specific root length is experiencing significant decreased selection, and there's no significance for selection on root to shoot ratio. But what we're interested in really is what form mycorrhizal mediated selection is taking. And so to get at this, we can assess the interaction terms which combine plant traits and fungal traits and allow for the assessment of coevolution. And today, in the interest of time, we're just going to talk about one fungal trait, fungal abundance. But I have many more fungal traits, so if you're a mycologist or really just interested in this work, come see me. And unfortunately, if we just look at fungal abundance, we have no significant selection that's being undertaken from the mycorrhizal's point of view. So if we take it all together, we see that these above ground traits are experiencing a positive selection event, whereas the root to shoot and specific root length are really experiencing negative. And none of this selection, if we think about it in terms of fungal abundance, is being related to mycorrhizals themselves. So if you're interested in interactions between plants and their microbes, I am starting an assistant professor position at Wright State in Dayton, Ohio in a few weeks, and I would love to talk to you. Either you can get a hold of me with any of these contact informations or come find me at the meeting. I'm really short. Talk to you later.